a bit of time talking to you about ImageLab software. This is our next generation imaging software to be able to drive two of our imaging instruments currently, which is the GelDoc XR Plus and the ChemiDoc XRS Plus. But more importantly, the software allows you to analyze images from any of our imaging solutions that are all driven by Quantity 1. So this reads any Quantity 1 file as well. So very nice, fully packaged next generation imaging software for band analysis. So if we look at the software itself and we look at the start page, so this clearly looks much different than Quantity 1 software. When you open the software, you get a nice start page here, which allows you to drive the instruments that the software currently runs, which is as I mentioned, the XR Plus and the XRS Plus. Along the left side of the work area, you have all the imaging analysis tools. So let's start by running an instrument. So if we click on New Protocols here, you get the protocol setup window. Now what's really cool about the software is that it has simple acquisition settings and analyze image settings and report settings that you can use to automatically analyze and acquire images. I'm going to start by just looking at the gel imaging settings. So that's here, gel imaging. And if we click select application, which is the first field under the gel imaging settings, we can see that now the software is entirely application driven. So just by clicking on a particular application, let's say an ethidium bromide stained agarose gel, the software automatically sets the instrument up for the light source, the filtering, and the light source and the filtering, and how to place the image. So that's ethidium bromide. There are, protein gels with the white transilluminator, again with the filter settings, and then for blotting, same sort of deal. Okay, so you get the filtering, low illumination, and camera settings as well. Everything is automated, so it makes it very straightforward for people to use who have never used an imager before, to just walk up to the instrument, click on the application they want to use, and they're ready to go. So let's assume that we're doing an ethidium bromide stain gel as an example. So we click there. The next step is image area. This is an option that we don't really need to worry about setting. You can basically, the camera will automatically focus for particular image areas that you set in the software, either for Bio-Rad gels or for your own custom gels. I typically skip this step and just go straight to exposure. Under exposure, you can get the software to automatically detect intense bands or faint bands, or you can manually set the exposure time as well. So it's really up to you. I typically set it for faint bands initially, and then I see how it looks. Once these settings are done, you can set, this is a protocol that we're setting up, so we can actually save this as a protocol here. And it will set it up. You can save it as a protocol file, which you can then call up time and time again in your protocol editor. Once I've set up the protocol, I can click, and I also like to have display options under highlight saturated pixels, just to make sure that I'm able to see whether bands have been over-detected by the camera. They'll appear red, and that means they're not quantifiable. Then I click position gel, and the software automatically controls the iris and the focus for you. So the only thing you need to worry about is positioning the gel under the camera. When you click position gel, it automatically turns the lights on, the white lights on the instrument, no UV, so it's very safe, no worries. And you can just slide the zoom, and it keeps the focus of your gel under the camera. So it's very nice in that regard. Now this, of course, is a previously taken picture because I'm in simulation mode of an ethidium bromide stain gel. But if you were to look at that gel under the white light, of course you wouldn't see the bands. But you'd see the gel, and it would always remain in focus. So you don't need to worry about writing little words on blots or gels or putting a ruler beside. The focus is automatically organized. Then you click Run Protocol, and you get your image. And it's that simple. So just to show you one last thing with respect to taking an image, because the only different setting is really with blots for chemi. And if you go down here, instead of having viewing for intense or weak bands, we have a signal accumulation mode. If you click Setup here, you can now set it up for multiple exposures. So let's take the first exposure of our blot at 1 second, the last exposure, let's say, at 10 seconds, and you can set the total number of exposures. So this will take 3 consecutive exposures, where the first exposure will be for 1 second, the second exposure, let's say, around 5 seconds, and the third exposure will be for a total of 10 seconds. And you click OK. Then we run the protocol, and you can see that it's actually taking exposures consecutively from 1 all the way up to 10 seconds. And if we click on each one, you can see the bands are getting darker in the image. You can see exactly what the exposure time for each of these images was. And if you want to save them, you just right-click. You can save individual ones or save them all. And you also have a nice image transform here, so you can see if you can see the faintest bands. And if not, then you can try going for a longer exposure time. If I'm happy with this particular image, and basically what I'm looking for is the image that has the most intense bands that are not red, not saturated, so I can quantify them. Click Select and Continue. I have my image, and I'm ready to analyze. It's as simple as that to use this software for driving the GelDoc XR Plus and the ChemiDoc XRS Plus.